What we were trying to investigate through this lab is how much the concentration of the glucose would affect carbon dioxide production through fermentation. Fermentation is an anaerobic process that is used to produce energy. In this case, yeast was the thing that was fermenting the glucose, and it only happens if no oxygen is present. Our hypothesis was that if we used higher concentrations of molasses, then we would get more carbon dioxide from fermentation. For this lab, we used some basic lab stuff like beakers and test tubes, some balloons, yeast, and molasses. And basically what we did was we put the yeast solution in a large beaker, and then we used the test tube for molasses.
Brandon, what are you doing?
He's doing it awful.
That looks really interesting.
Oh,
So exactly how much do you have to pour in?
5 million is
5
of yeast.
milliliters of yeast.
How much
So
is
this
that?
is yeast?
Yeah.
And
much
what's
is
the
that?
liquid?
Well, it's a yeast solution.
And so how much of this do you need?
5 milliliters.
What are you going to do with it?
Move this. Good job, Sean.
What are you going to do with it?
We're going to add this to the test tube, and then we're going to add water.
So what exactly are you doing right now?
We're mixing our solution of water, yeast solution, and molasses.
So how particularly are you mixing it?
We're using 12.5 milliliters of water, 12.5 milliliters of molasses, and 5 milliliters of yeast.
So are you like shaking it around? Or
No, we're just inverting it.
If you go all the way to the top, then you're good. Now, you want to put the lip of that one over the top of that lip, and then you want to tilt the big one until the water meets the other water. And then the second that happens, you flip it.
Okay. Don't do it over the table. I got this.
Just do it over the table. Graham's got it. Yep. And you want to maybe angle the small one a bit more.
Okay, Eddie.
So then once they meet, oh, way better than Dylan. Okay. Get
So
it.
Sungmi, what are these balloons? What are they for?
Oh, the balloons will catch all the gas that is made, and it'll show more than the miniature test tube will. And as the gas is filled into the balloon gets bigger. And then in the end, we'll measure the circumference of the balloon.
And so you're comparing it with the other ones as well.
Yes.
So what are you guys doing?
So we take this, and we go like this,
Go like that.
and then we wrap it around, yeah. You wrap it around.
Make sure you get
Having
the
trouble
words
with
there.
And so now we know that right here,
There you go.
so we know that this is the distance that
Upper right there, yeah, right eyeball.
yes. And then you put it like this, and then you measure it with a little ruler.
The data for this experiment was measured in the size of the balloon in cubic centimeters. The results showed that concentration differences did not make a measurable difference as far as the results variation goes, but there seems to be a very, very small, almost undetectable positive correlation between the concentration of molasses and the amount of carbon dioxide produced, but the data is not solid enough that we can draw an actual conclusion from it. But this is clearly caused by variation and error, and it could have been caused by inactive yeast, tight balloons that were difficult to inflate, different treatment by the groups of the balloons prior to testing, whether or not oxygen was actually missing from the testing environment, and just human error.